to the continuation class of fertilization. In the previous class, we have learned about the different events of fertilizations, where we have learned about the formation of sperm, ovum, their fusion, leading to the formation of a zygote. Here, so today we'll just look at the sex determination of the zygote. Right? What will be the sex of the zygote, or whether the zygote will develop into a male sex or into a female sex. So just briefly, we look at topic sex of the child. Now we all know that we, human beings, we have got 46 number of chromosomes, right? We have got 46 number of chromosomes. Out of 46, 44 are the autosomes or the body chromosomes, and the remaining 2 are the sex chromosomes, which are different in case of male and in case of females. So, regarding the sex chromosomes, it can be XX, like in female, or it can be XY in male. Clear? So now we will discuss the type of gamete formation from each of the parents, which will result in the formation of a particular sex of the child. So let's write down, since in case of human being, the reproduction is of sexual type, that means it involves 2 parents. So we'll write over here the female parent and the male parent. The chromosome number of the female sex chromosome is XX in case of female, and in case of male, the sex chromosome is XY. Clear? So the sex chromosome of female is XX and the sex chromosome of male is XY. Y denotes the male chromosome. Now, there must be the formation of gametes. Now, during gamete formation, the type of cell division that occurs over here will be meiosis, which is called reductional division. Clear? So the type of cell division that occurs during the formation of gametes will be meiosis cell division, which used to call the reductional sex or reductional cell division, where the number of chromosomes in the gametes will be haploid. So when we write down the type of gametes formed from the female, then therefore we'll find one X chromosome, then we'll find another X chromosome. That means the female is homogametic. Clear? Why do we call it homogametic? Because both the chromosomes are same. So therefore the gametes, that is ova, which is produced by the female, it always bears X chromosome. Clear? And gametes are haploid. Clear? And gametes are haploid. Now, similarly in case of male, the same type of cell division, that is meiosis, occurs, and therefore the gamete, that is sperm, which is produced, bears one X chromosome and it bears Y chromosome. So it can be either X or Y. So this is the sperm produced by the male parent. So the ova always it bears X chromosome, whereas the sperm, it contains X chromosomes or Y chromosomes. Why? Because the male is heterogametic. There are X chromosome and Y chromosome. Clear? So the sperm that is produced by the male bears X chromosome or it bears Y chromosome. So these are the types of gametes which are produced. Now after the formation of gametes, the gamete has to be fused, which is called fertilization. Clear? And remember, gametes are always haploid. So ova has always X chromosomes, and the sperm has X chromosomes or Y chromosomes, and they are also haploid. It bears half the number of chromosomes, that is 23. Now, after the formation of gametes, the gamete has to be fused, which is called fertilization. Now, what is fertilization? It is the fusion of gametes, that is the ova and the sperm. Okay. So during fertilization, let's say, if the ova bearing X chromosome, if the ova bearing X chromosome will fuse with the sperm bearing X chromosome, then therefore the resulting zygote will be female. Clear? Similarly, if the ova bearing X chromosome fuses with the sperm bearing Y chromosome, then resulting zygote will develop into male. Clear? So always when we look into the cross, so always there is 50-50 chance of development of a male child or a female child. So this will be XY, it will be male. So look over here. So what we are finding over here is that the sex of the child, that is male or female, is dependent on the type of sperm that will fertilize the ova. Clear? So if the sperm bearing X chromosome will fertilize the ova, then the resulting sex of the child will be female. So over here, the type of sperm is helping in the determination of sex of the zygote. Similarly, if the sperm bearing Y chromosome will fuse with the ova, then the resulting sex of the zygote will be male. That is, the zygote will develop into the male sex. Clear? So here we can say that the sex determination is dependent on what type of sperm will fertilize the ova. Because ova or the egg, which is the female gamete, it always bears the X chromosome. Whereas the type of sperm that will help in sex determination, because sperm carries X chromosome and Y chromosome. Clear? So remember carefully that the sex of the child is dependent on the type of sperm that will fertilize the ova, and therefore the respective zygote will either develop into the male sex or into the female sex, depending on whether it is fertilized by the sperm bearing X chromosome or by the sperm bearing Y chromosome. Clear? So by this way we have finished the sex determination of the child. Now, after fertilization, we have learned that there will be formation of zygote. Clear? Because zygote is a single cell which is diploid, having 46 number of chromosomes. It's a single cell. Now that single cell has to undergo repeated mitotic cell division, leading to the formation of an embryo, we can see. Clear? So now in the next topic, we will learn about embryogenesis, the process which will lead to the formation of an embryo from a single fertilized egg or from the zygote. And we will see what are the different events under embryogenesis that will lead to the formation of an embryo and lead to the formation of a fetus. So now we will discuss about the topic embryogenesis. What is embryogenesis? Embryogenesis means development of the embryo. So you can see over here, embryogenesis means formation. Clear? So embryogenesis is the process of formation of embryo from a fertilized egg. So when the egg will be fertilized, when the egg will come in contact with the sperm, it will lead to the formation of a zygote. Now that zygote will develop into the embryo. So what is embryogenesis? It is the process where there will be formation of an embryo from a fertilized egg. Clear? Now we will see the type of events that occurs during embryogenesis. So therefore, embryogenesis has the major stages. Now, what are the stages? First stage is cleavage. Stage of rapid cell division, that is mitotic cell division. Remember, zygote is a single cell which is diploid. Now that single cell has to undergo repeated mitotic divisions, which we'll discuss afterwards. Then from cleavage there will be formation of modula. Then after that modula stage, we will find the second one is called blastocyst formation. Then third stage is implantation, in which the blastocyst will be implanted in the wall of the uterus. Then next one is gastrulation, where there will be formation of embryonic germ layer. The last one is organogenesis, where there will be formation of organs. So these are the major steps of embryogenesis. Alright, so we will go through each and every step in detail. Don't worry for that. Now let's see over here. So this is the structure of a fallopian tube. This is the ovary, and this is the part of the uterus. You can see over here. So this is the uterus, this is the fallopian tube, and this one is the ovary. Now from the ovary, there will be the release of ova or egg during ovulation. Clear? During ovulations, the secondary oocytes will be released from the ovary, and that secondary oocytes will be collected by the fimbriae. Do you remember what is fimbriae? The finger-like projections which are present on the broad end of the fallopian tube or of the oviduct. So those fimbriae will collect the ova released from the ovary. Now, this fimbriae will collect the ova or the secondary oocytes, which will enter now inside the fallopian tube. So you can see over here the marking A. So this is the structure of the secondary oocytes. Clear? Which is surrounded by the corona radiata, the cells of follicles or follicular cells. So this is the secondary oocytes. Clear? Now, these secondary oocytes, when it's reaching to the ampulla isthmus junction of the fallopian tube, it will lead to fertilization when it comes in contact with the sperm. So you can look at the figure number B. The secondary oocytes is coming in contact with the sperm. So this is the event of fertilization. Clear? So the sperms are coming in contact with the ovum or with the ova, leading to fertilization, and thereby it forms the zygote. Alright, so all these events are occurring in the fallopian tube. Remember, the event of fusion of sperm and the ova is occurring in the ampulla isthmus junction of fallopian tube. Now, after fertilization, there will be formation of zygote, a single cell which is diploid. Clear? Now, that zygote undergoes repeated mitotic divisions. That means the number of cells keeps on increasing from 1 cell to 2 cells, 2 to 4, 4 to 8, 8 to 16, 16 to 32. So it keeps on increasing in cell number, and those daughter cells which are produced, they are called blastomere. Clear? So the zygote will divide. A single cell zygote will divide into 2 daughter cells. You can see over here. First cell, second cell. Clear? So this is the 2 cell stage. All these events are occurring inside the fallopian tube. Now again, these 2 cell stages will again divide into the 4 cell stages. In between these 2 to 4 cell stages, we will find a short period of 3 cell stages. Only for a short period of time. Again, it will divide into 4 cell stages. And you can see over here, it's a type of unequal division, because you are finding the 2 larger cell and the 2 smaller cell. Clear? So zygote single cell will divide into 2 daughter cells or 2 cell stages, 2 into 4 cell stages. Again, 4 cell stages will divide into 8 cell stages. Clear? And 8 cell stages will again divide to form 16 cell stages. So you can see over here the 16 cell stages. Again, this 16 cell stage will divide into 32 cell stages. So therefore, the cell with 16 blastomere or with the 32 blastomeres, or we can say in 16 cell stage or in 32 cell stage, are known as Modula. M-O-R-U-L-A, Modula. Clear? So that means a zygote, after the fusion of gametes, when the zygotes will be produced with a single diploid cell, it will undergo repeated mitotic divisions to form the 2 cell stage, 2 to 4, 4 to 8, 8 to 16, 16 to 32. So this 16 to 32 cell stage is known as Modula, and all these events are occurring in the fallopian tube. You can see over here A, B, C, D, E. So A means the ovum, B indicates the process of fertilization, C means that zygote has undergone mitotic division leading to formation of 2 cell stages. Now from 2 it forms the 4 cell stage, that is D, then E, and F is the morula with 16 cell states or with 32 cell states. Now after the formation of morula, it enters inside the uterus. Clear? It enters inside the uterus. Now there are certain processes, alright, where the morula will change into blastocyst, which we'll discuss, don't worry. So morula will change into blastocyst. You can see over here H, and after the formation of morula, it will come in contact with the endometrium of the uterus. Clear? So blastocyst will come in contact with the endometrium of the uterus, and when it comes in contact and will attach with the wall of the uterus, it is called implantation. Clear? And after implantation, there will be gastrulation, where there will be formation of embryonic germ layer, ectoderm, endodem, mesoderm. Clear? And after gastrulation, finally there will be formation of the organ, which is called organogenesis. Clear? So the structures where the entire events of embryogenesis occurs. Clear? So remember, ovary will release the secondary oocytes, which will be collected by the fimbriae, and from fimbriae will go into the fallopian tube, where there will be fusion of gametes, leading to the formation of zygote. Zygote, which are diploid single cell. It undergoes repeated mitotic divisions called cleavage, from 1 cell stage to 2 cell stage, 2 to 4, 4 to 8, 8 to 16, 16 to 30 cell stage, which is called morula. So all these events are occurring in the fallopian tube. After the formation of morula, it enters inside the uterus. Then this morula will change into blastocyst. The blastocyst will come in contact with the endometrium lining of the uterus, which is called implantation. After implantation is over, the next event over, which is called gastrulation, where there will be formation of embryonic germ layer, and after gastrulation there will be organogenesis, which is the process of formation of organ. Clear? Now we will go through these steps one by one. Clear? So first of all, we will begin with the process, the first event called cleavage. Now what is cleavage? Cleavage is a repeated mitotic divisions in the zygote, which transforms a single fertilized egg cell into the hollow spherical and multicellular blastula. Now what am I trying to say over here? Listen very carefully. Cleavage means is the process of repeated mitotic cell division. Mitotic means mitosis. Clear? So there will be rapid mitotic cell divisions where the number of cells keeps on increasing. We have learned in the previous diagram that from a single cell zygote it divides into 2 cell stages, 2 to 4, 4 to 8, 8 to 16. So all those divisions which are occurring is simply the mitotic cell division. Clear? And remember one thing, only the number of cells are increasing. Alright, the size of the cell is not increasing. The size of the cells will be similar to that of the zygote, but only the number of cells are increasing in number. The size will remain the same. The zygote is still surrounded by the vitelline membrane. It is surrounded by the zona pellucida layer, and finally it is surrounded by the corona radiata cells. Remember this thing. Clear? Zygote will find all the 3 layers. We will find the vitelline membrane, we will find the zona pellucida, right? We will find the cells of corona radiata. That means the size of those cells will remain the same during cleavage. Only the number of cells keeps on increasing due to repeated mitotic divisions. And this particular cleavage occurs from a single fertilized egg cell. Clear? Till where it will form the blastula, which is multicellular. So multicellular means it is composed of many cells. Clear? So from a single cell, from a single zygote, it will keep on dividing continuously by mitotic divisions till it will divide until it will form the multicellular blastula. Clear? Now, second thing, it increases cell number. Those daughter cells which are produced by the mitotic cell division, they are called blastomere. Keep in your mind. Clear? Those daughter cells which are produced by the repeated mitotic divisions,
from a single fertilized egg, they are called blastomere. So it just increases the cell number. It just increases the number of blastomere. Alright, 2 cell stage, 4, 8. So that means the number of blastomeres keeps on increasing. And the resultant blastomere becomes progressively smaller, which is called compaction. That means those blastomeres which are produced, if you look over here in the previous figure, come back to over here, come over here. If you look over here, the number of cells over here, 2 cell states, is a little bit larger. 4 cell states, 2 larger and 2 smaller. But when this division keeps on going on and on, the resultant blastomeres will be smaller. And this particular process is called compaction. So those blastomeres which are produced by repeated mitotic divisions during cleavage undergoes compactions, where we will find the smaller blastomere and we will find the larger blastomere. Clear? Now next point. There will be increase in the nuclear cytoplasmic ratio. Why? Because there will be rapid DNA synthesis. Now why there is need for rapid DNA synthesis? Because those new blastomeres which are produced also must contain DNA and the chromosomes. So there has to be rapid DNA synthesis to ensure that the chromosomes can be distributed in those newly formed blastomere. So there will be rapid DNA synthesis, but there is no cytoplasmic synthesis. Only the new DNA will be synthesized, but there will be no synthesis of cytoplasm. So therefore there will be increase in nuclear cytoplasmic ratio. Next one, interface in cleavage division is short and does not involve growth. Now what is interface? Interface is a stage of cell division. We have learned in class 11 about the process of cell division. So mainly in cleavage, the interface is usually very short. So this is mainly about the idea about what is cleavage. Now we will see the cleavage in case of human zygote. Clear? So let's go to the next slide. We'll look at the cleavage in the human zygote. Clear? So you can see over here, this is the zygote. You can see over here the cell membrane, the vitelline membrane. Alright, this piece is called perivitelline space. This is called the layer called zona pellucida, and it will be covered by the corona radiata cells, which is missing over here. Okay, so we can say that over here we will find the cells of corona radiata. Alright, now let's see the zygote. So about 24 hours after fertilization, zygote divides into 2 blastomeres. That means a single cell zygote. Alright, listen very carefully. A single cell zygote which is diploid to N. It will divide into 2 blastomeres. You can see over here 1, 2. So these are the 2 blastomeres. Clear? So a single cell zygote will divide into 2 blastomeres within 24 hours after fertilization. That means after 24 hours of fertilization, a single cell zygote will divide into 2 cell stages. How many blastomeres? 2 blastomeres. So this particular stage or this particular division is occurring within or about 24 hours after fertilization. Next one. Next cleavage takes place within 40 hours after fertilization. Now why are we saying that the next cleavage occurs? Because cleavage is a repeated mitotic divisions where the cell or the zygote, the single cell has to divide on continuously to form a 16 or 32 cell stage called modula, which has to finally change into the blastocyst. So again after the next cleavage takes place within 40 hours after fertilization. That means from the 2 cell stage it has to form the 4 cell stage. So you can see over here this is the stage with 2 blastomeres. Again these 2 blastomeres will divide into a cell containing how many blastomeres? 4 blastomeres. 1, 2, 3, 4. Again this particular cell containing 4 blastomeres will divide, will undergo mitosis to form the 8 blastomere stage. So single cell will divide into 2 cell stage, 2 cell stage into 4 cell stage, and 4 cell stage into the 8 cell stage. Again, this 8 cell stage will divide into 16 cell stage. Clear? So here the repeated mitotic divisions occurs after fertilizations. Next one. Third division occurs about 3 days after fertilization. The next one, subsequent divisions one after another occurs, resulting in the formation of a solid mass of cell called modula. That means a single division keeps on occurring, repeated mitotic divisions keeps on occurring till the single cell zygote will form the blastocyst. But first of all, remember, within 24 hours fertilization, this will divide into 2 cell stages. Then 40 hours of fertilization will divide into 4 cell stages, and nearly after 3 days there will be subsequent mitotic divisions which will lead to the formation of 8 cell stages, and these divisions keeps on occurring till it will form the 16 cell stage or 32 cell stage called morula. Clear? And finally, over here, during this period, embryo slowly moves downward toward the uterus from the fallopian tube. That means when this stage is going on, when the zygote is undergoing cell divisions, alright, slowly it's moving from the fallopian tube towards the uterus. Clear? So one more time. It is a human being, so about 24 hours after fertilization, the zygote is single cell, it will divide to form the 2 cell stage, that is blastomere. Again, the 2 cell stage blastomere will again divide to form the 4 cell stage. Clear? And again, that 4 cell stage will divide into the 8 cell stage. So this particular division keeps on occurring till this 8 cell stage blastomeres will divide to form a 16 cell stage called morula. M-O-R-U-L-A, morula. So morula is a hollow solid mass of cells with either 16 cell stage or with 32 cell stage, and the resulting cells are called the blastomere. Alright, keep in your mind, the resulting data cells are called the blastomeres. Clear? And those particular cells with 16 to 32 cell stage or with blastomeres, they are called modula. Now we will see what is modula. Look over here. Modula is a solid ball of 16 to 32 blastomeres. So it will be with 16 cell stage or it will be with 32 cell stage. So this is in fact the 16 cell stage. Again, this particular modula with 16 cells will again undergo mitosis cell division to form the 32 cell stage. Clear? So that means a cell with 16 blastomere or with 32 blastomeres, they are called modula, which is a solid ball. Okay, next one. Now, after the formation of modula, listen carefully. These particular stages are occurring inside the fallopian tube. That means they are slowly moving from the fallopian tube towards the uterus. So now, this modula, it reaches the uterus by the end of the 4th day. So at the end of the 4th day, this modula will reach the uterus. Next one. Modula is also surrounded by zona pellucida. So you can see over here the renal cells, or we can say Madeline membrane, we can say over here. This layer is called the zona pellucida, and outside the zona pellucida we'll also find the cells of corona radiata. So in fact, morula contains the zona pellucida layer and also the corona radiata. Clear? Next one. It is about the same size as zygote. That means the size of the morula and the size of the zygote will be same. Why? Because we have learned that during mitotic cell division, alright, only the number of cells keeps on increasing. The number of blastomeres keeps on increasing. There, the size remains the same. So therefore, till the time it will form the morula, the size of the morula will be same. Is that of the zygote. There is no such difference in the size of the zygote and the morula. The difference only lies with the number of blastomeres. Because in case of morula, there will be 16 blastomeres or there will be 32 blastomeres. But a zygote will only contain a single cell. Clear? So difference lies with only the number of blastomeres. When we compare the morula and the zygote, the size remains the same. Next one. It consists of an inner cell mass and the trophoblast or the trophoectoderm. Now which one is the inner cell mass and which one is the trophoblast or the trophoectoderm? Okay. Now those single, or let's say those smaller cells, like let's say in the 16 cell stage or in the 32 cell stage, those cells which are smaller, alright, those cells which are smaller, just draw over. Those cells which are smaller, let's say the layer called zona pellucida. Alright, we have got pattern membrane over here. No, don't worry, it's the pattern membrane. And we can have over here corona radiata. Clear? Now those smaller cells which are present over here, they will form the outer layer like this. So this particular layer is called the trophoblast or the trophoectoderm. And inside that one we will find the larger cells, you can see over here, which is called the inner cell mass. Okay, so morula is mainly composed of two types of cells. First one is the inner cell mass, which is present in inner sides, and those outer lining, which is called the trophoblast or the trophoectoderm. So these are the trophoectoderm. So you can see over here also. So those particular layer of cells, these are the trophoectoderm or the trophoblast, and these are the inner cell mass. Clear? Alright, what is this one? This is the trophoectoderm or the trophoblast, and this one is the inner cell mass. Now we will learn about the formation of blastocyst from the morula. Clear? So before that, we will do a quick revisions. We will try to recall what we have learned till now. So till now we have learned that from the embryo there will be release of secondary oocytes. Clear? We are calling it as secondary oocytes, not as the ovum. Or those secondary oocytes will be collected by the femore of the fallopian tube. So this one is the structure of the ova. The point number A. It's surrounded by the cells of corona radiata. These are the cells of corona radiata. We have done the structure of the ovum. Then this ovum will be fertilized by the sperm. This is point number B. Fertilization occurs in the ampulla stomach junction of the fallopian tube. Now after the fusion of male gamete and the female gamete, there will be formation of zygote. Yeah. Now that zygote undergoes cleavage or repeated mitotic cell divisions, where a single cell zygote will divide into a two cell stage with two blastomere. Clear? Again, that two cell stage undergoes second mitotic division to form a four cell stage with two smaller blastomere and with two larger blastomere. Clear? Now again, this four cell stage will divide into 8 cell stage. 8 cell stage will again divide into 16 cell stage, and this will divide into 32 cell stage. So a blastomere with 16 or 32 cell stage is known as modula. So you can see over here, so this particular one, the point E is the modula, is slowly moving from the fallopian tube towards the uterus. Clear? Now, this modula now will change into blastocyst, which we will discuss now. So modula now changes into blastocyst. Now blastocyst will come in contact with the endometrium of the uterus. Now the endometrium is the inner lining of the uterus, which is highly convoluted, highly glandular due to the action of the hormone progesterone and estrogens. So blastocyst will come in contact with the lining of the uterus, that is endometrium, After that, there will be implantation, and after implantation, there will be formation of 3 embryonic germ layer, which is called gastrulation. And after gastrulation is over, there will be formation of organ, which is called organogenesis. Now we learn about the formation of blastocyst from the modula. Clear? So keep in your mind that modula is a solid mass with 16 blastomere or with 32 blastomere, and it consists of the inner cell mass and the trophoectoderm or the trophoblast. So listen over here. Blastocyst formation from modula. So when the modula comes in contact with the uterine wall, that means when the modula enters inside the uterus, the glands of the uterine wall, it will secrete a glycogen-rich fluid called the uterine milk. Clear? Now this uterine milk, it is collected between, or it enters between the trophoblast and the inner cell mass of modula, leading to the formation of a cavity called blastocyst cavity or blastocele. One more time. This uterine milk, which is secreted by the glands of the uterine wall, when the modula comes in contact with the uterus, this particular uterine milk, it enters between the inner cell mass and the trophoblast of the modula, forming a cavity called the blastocyst cavity or blastocele. Clear? Now, with the appearance of blastoceles, modula now changes into blastocyst by the 5th day after fertilization. So that means when there will be formation of blastocele, alright, in modula, now modula will change into blastocyst, and this particular changes occurs by 5th day after fertilization. So let's see the structure of blastocyst. So look over here. The structure of modula, which is now being changed into the blastocyst, and the process is called blastulation. So you can see over here, modula still contains the zona pellucida layer. It contains the corona radiata cells, which slowly starts degenerating. This is the cells called the trophoblast or the trophoectoderm, and this is the inner cell mass. Clear? This is the inner cell mass, and this is the trophoectoderm. Now what we have learned is that when the modula enters inside the uterus, when it comes in contact with the uterine wall, the glands of the uterine wall will secrete the glycogen-rich fluid called uterine milk. Now that uterine milk, it enters in between the trophoblast and the inner cell mass, and that will form a particular cavity which is called the blastocele. So with the appearance of blastocele in the modula, now modula will change into blastocyst. Okay, so look over here. So at the end of the 5th day of fertilization, blastocele enlarged. That means this particular cavity, the blastocele which was formed, now starts enlarging, to enlarge in size, and pushes the inner cell mass to one side. That means this blastocele, this cavity will enlarge. Due to the enlargement of the blastocele, it will push the inner cell mass to one side. You can see over here, this is the inner cell mass which has been moved to one side. Clear? So in fact it was in the center. But when the cavity keeps on increasing, keeps on enlarging, those blastomere or those inner cell mass will move to one side, thereby forming a knob called the embryonal knob. So this particular knob is called the embryonal knob, which is formed due to the enlargement of the cavity called the blastocele. So when the blastocele enlarges, the inner cell mass will be pushed to one particular side, forming a knob which is called the embryonal knob. And this embryonal knob, listen carefully, this embryonal knob, which is composed of inner cell mass, this will give rise to embryo. Clear? So this particular knob is called the embryonal knob. So we can see over here, this one is called the knob, which is called the embryonal knob, and this embryonal knob will give rise to the embryo. Clear? Second one, Trophoblast divides to form the extra-embryonic fetal membrane, amnion and chorion. That means this trophoblast is not involved in the formation of embryo. The inner cell mass are involved in the formation of embryo, and this trophoectoderm or the trophoblast, they will form the fetal membrane called the amnion and chorion. Clear? So how to identify the blastocysts? By the appearance of the cavity called blastocyst cavity or the blastocysts. So when modula enters inside the uterine cavity, uterine walls are producing uterine milk. That uterine milk enters in between the trophoblast and the inner cell mass, forming a cavity called the blastocells. These blastocells, once they are formed, then modula will change into blastocyst or the blastula, and this cavity enlarges, pushing the inner cell mass to the one particular side, forming a knob called embryonal knob, which will give rise to the embryo, and this trophoectoderm or the trophoblast, they will divide to form the amnion and chorion, and will occur after the 5th day of fertilization. Clear? So till now we have learned about the first stage of embryogenesis, that is cleavage or process where there will be repeated mitotic divisions. Then we have learned about the process of formation of modula, a solid mass of cells with 16 or 32 cell states, right? Then we have learned about the formation of blastula from the modula. Clear? Now this blastula, it goes and will be implanted in the wall of the endometrium lining of the uterus, which is called implantation. Then there will be gastrulation and organogenesis, which we will take in the next class. Yeah, so in the next class we will learn about implantation, we will learn about gastrulation, and we will learn about organogenesis. Yeah, okay, thank you.